Okay, this is back half of chapter 28 on pregnancy and human development. And what we're going to do is walk the rest of the way out from embryogenesis through fetal development to the neonate to birth. So organogenesis is an important milestone because it is the dividing line between embryonic and fetal development. Gastrulation generates the three germ layers that are going to contribute to all the organs and organ systems in the developed fetus and neonate. At the 8th week, the organ systems become recognizable, and that's the end of the embryonic period. The embryo began as an embryonic disc that at first had 2 cell layers and then 3, right? Ectoderm, mesoderm, and endoderm. And then what will happen is that this sheet, this disc, will begin to fold both laterally and from front to back, okay, in order to generate the body cavities and the body chambers, okay. The primitive gut is formed from an endometrial fold, and that forms the epithelial lining of the GI tract where it's simple epithelium. Organs of the GI tract become more apparent and oral and anal openings appear. The mucosal lining of the respiratory tract forms from pharyngeal endoderm, and glands appear further along the tract. So what you're looking at here is what we're talking about, right? This is the disc. This is you, okay? Amnion, yolk sac. Here we're taking a section of the disc, and what you're seeing here is the folding that goes on laterally, okay, in addition to the folding that goes on from head to tail, right? So you can see the arrows show the direction of the folding, right? So we're folding inward here, and we're folding downward here and here, right? What'll happen through the course of the weeks of embryonic development is that you'll begin to see along the dorsal side structures called somites. The more somites you have, the further along in the developmental process you are. And the somites mark the segmentation of the body and will eventually contribute to the vertebral column and will enclose the spinal cord, okay? But what they do is they contribute to the formation of part of the axial skeleton. They contribute to the formation of musculature in the body wall, and they will enclose the ectodermal tissue that's going to form the spinal cord, the brainstem, and the brain. Okay, if we take a sagittal section through this, you can see here there's the foregut, right? There's our yolk sac. So this is top, this is bottom, this is head, this is tail. This is the neural tube, okay? And this is where the brain and the brainstem are going to be. And then the notochord down here is what actually induced the formation of the neural tube, which drops below the ectodermal layer on the dorsal side and will await for the development of the somites to develop structures that will enclose it, okay, in addition to generating the other tissues that we discussed. You can see here the digestive system, right? This is going to be the oral cavity. That's going to be pharynx. That will become esophagus. This is stomach, okay? This is going to be liver and pancreas. This is going to be gallbladder, liver here. And this is going to be small and large intestine, okay? And then rectum and anus will come out down here, okay? Right by where the tail is, right? This is umbilical cord, okay? And this is our connection to the yolk sac here, okay? So this is eventually going to be the umbilicus of the fetus, okay? In addition, you can see other internal cavities forming, primitive respiratory system with what's going to be the trach and the lungs, okay, beginning to form. You've got the thyroid gland coming off here, okay. So the bulk of this lining is endodermal, okay. The only part that's going to be mesodermal is going to be the connective tissue that lies beneath the endodermal, what will eventually be the epithelium, okay, and any smooth muscle, all right, and of course the surrounding membranes, okay. Neurulation is the first major event of organogenesis and is going to produce the brain, brainstem, and spinal cord. The notochord induces this, generating a neural plate, which is a thickening over the dorsal side of the embryonic disc that will eventually become the neural groove. And the neural groove will then close up from basically about halfway down the long axis of the embryo and proceed towards the caudal and the rostral end, right? The caudal end is the end where the spinal cord would terminate, and the rostral end towards the nose is where eventually the brain is going to develop. And all of this is induced by chemical signals that come from the notochord. By the 22nd day, the neural folds are going to fuse into the neural tube, okay? And those are the structures that will generate. The last thing to fold up is going to be the brain, okay? So again, all of this starts out as an open groove along the dorsal side, and then we fold up from middle towards the end and from middle towards the head, and the last thing to close up is the brain. And again, this mimics the evolution of the nervous system in evolution, right? Where the brain was the last thing to show up on the evolutionary tree, specifically the cerebral cortex. Before the neural tube fully forms, there are cells that are in the ridges that will eventually fuse and form the top of the tube called the neural crest, and they migrate all around the embryo and contribute to cranial and facial bones, to spinal and sympathetic ganglia and nerves, portions of the adrenal medulla, the melanocytes, and certain connective tissues, okay? But basically, the neural crest cells are, they sort of follow their own developmental path, okay? So they're a subset of components that are originally from ectoderm, okay? You get brain waves at the end of the second month, okay? First evidence of mesoderm is the appearance of the notochord, which will eventually become the vertebral column, okay? It will contribute to the vertebral column. The mesodermal aggregates lateral to the notochord are the somites, the intermediate, and lateral plate mesoderm, okay? So somite is just a it's those little segments of tissue along the dorsal side. The more somites you have, the further along the developmental pathway you are, all right? Then the intermediate mesoderm and the lateral plate mesoderm are going to form muscle and bone and other connective tissue, okay? The somites have three portions. The dermatome forms the dermis of the skin on the dorsal body. So this again indicates it's a mesodermal derivative because it's contributing to the dermis of the skin, which is connective tissue. The sclerotome generate the vertebrae and the ribs, and the myotome are going to form skeletal muscles in the neck, the trunk, and the limbs, right? And again, the names kind of tell you what they do. Dermis is referring to the skin. Sclera means hardened, okay? So that contributes to bone, and myo indicates muscle. Intermediate mesoderm is going to form gonads and kidneys. Lateral plate mesoderm has subdivisions, somatic and splanchic mesoderm, okay? Somatic mesoderm is going to form the dermis of the skin in the ventral region, parietal serosa of the ventral body cavity, the tissues of the limbs, while splanchic mesoderm is going to form the heart and the blood vessels. and most connective tissue in the body, and almost the entire wall of the digestive and respiratory organs, with the exception of the endodermal lining, which is the simple epithelia that line the gut and the lungs and the urogenital system. Okay? At the end of the embryonic period, bones ossify, skeletal muscles form and contract, and the kidneys begin to develop and the gonads form. Lungs and GI organs are going to form their final shape and be located in their final body position. Blood delivery to and from the placenta is effective, and the heart and liver are visible on the ventral surface. So you can see here, again, at 17 days, the situation, right? There is the neural plate. There's a notochord that induced the formation of the neural plate. There's our mesoderm, okay? And so we've got the yolk sac down here, right? We've got endoderm, okay, that is going to produce the linings of the gut and the respiratory and the GI, okay, as we fold in this direction, okay? And then we're going to, with this mesoderm, form those subdivisions that we talked about, right? So we have yet to neurulate, right? This is still primitive streak stage at 17 days. And then you can see the changes that take place, right? We generate the neural groove. You still got notochord here, but there we have, you see the populations of neural crest cells, right? But there's our lateral plate mesoderm, right? And then there's our somites, and then there's our intermediate mesoderm, okay? So this, the somite here, is going to form vertebral column and muscle, right? This is going to contribute to organs, as will the lateral plate mesoderm, okay? So this is at 20 days, so almost 3 weeks. And then after the 3-week period, the neural groove has become the neural tube, has dropped below the ectodermal layer. The neural crest cells have begun to migrate throughout the body and contribute to the structures that they are part of. Components in the heart, components in the skull, the adrenal glands, and so on, right? There's our somite. We still have notochord here, okay? And our lateral plate mesoderm, okay? The folding, direction of folding is shown here. That's endoderm right here, okay? This is a transverse section, just to keep you oriented, okay? And then at the end of week 4, things have changed quite a bit, right? There's the neural tube, but now we've become completely enclosed inside components from the somites, right? So what we have here is dermatome, there's sclerotome, there's myotome. So sclerotome here is going to form bone around the neural tube, okay? Myotome, the muscles of the body wall and the limbs, okay? And dermatome, the dermis of the skin, okay? Lateral plate mesoderm, you can see out here, limb buds, okay? Kidney and gonads from intermediate mesoderm. You can see the establishment of the peritoneal cavity, and now we have the alimentary canal established, right? With the lining of the gut being endoderm, okay? So a more complex body plan begins to emerge, right? We have now the beginning components of the dorsal and the ventral body cavities, okay? You can see here in this flow chart what forms what, okay? Ectoderm, mesoderm, endoderm, okay? It's a nice slide to sort of star and keep in your head, okay? Bottom line, the way I approach this is I memorize what the endoderm and the ectoderm form, okay? And then the mesoderm is everything else, all right? So I think that's probably the easiest way to approach this. Now, fetal circulation is unique because there's no source of oxygen coming from the air. So we're floating in amniotic fluid and our lungs are filled with it. The first blood cells come from the yolk sac by the end of the 3rd week. The embryo has paired vessels that form the heart that fuse together and bend into an S shape. The heart is beating by 3.5 weeks before it has blood to pump, okay? Modifications. umbilical arteries and veins, which are part of the umbilical cord, and then the vascular shunts, which include the ductus arteriosus, the ductus venosus, and there's also a hole between the two atria called the foramen ovale, and there is also going to be bypasses that feed blood into the umbilical cord that come off of the iliacs, right? All get blocked at birth and form ligaments, and that restores the normal circulatory pattern in the adult so that oxygenated blood is dealt with by the left side of the heart and deoxygenated by the right side. So the vascular shunts, the ductus venosus bypasses the liver and jacks into the inferior vena cava, so it brings oxygen and nutrient-rich blood into the inferior vena cava. The foramen ovale is the connection between the right and the left atria in the heart, and this helps to bypass the pulmonary circulation. And then the ductus arteriosus connects the aorta and the pulmonary trunk, and this allows us to bypass the pulmonary circulation and send the blood directly into the aorta. Okay, so you have to imagine now that we are in utero, right? We're inside the mother's womb. This is our placenta, okay? And watch the direction of blood flow, right? This is oxygen poor blood flowing down the vena cava, okay? And is going to enter the external iliac, right? And branch off of it, and then we form here the umbilical arteries, which carry deoxygenated and nutrient-poor blood away from the embryo. They work their way down to the placenta, where active and passive transport remove waste products and load in oxygen and nutrients and vitamins and minerals and antibodies, okay, from mother. And then the umbilical vein is going to bring this back, dump into the ductus venosus and into the inferior vena cava, and then we head up into the right atrium. And then as the two atria squeeze, the right atrium sends blood in two locations. It sends it into the right ventricle and also into the left atrium, okay? And so what happens next is the ventricle squeeze is the right ventricle sends the blood into the pulmonary trunk, but the bulk of that blood goes directly into the aorta through the ductus arteriosus. And then as the left ventricle squeezes, it sends the blood directly into the aorta. And so we bypass the pulmonary circulation, but we still get blood supplied to the structures that make up the lungs and the trach and the bronchi, because those have feeds off of the systemic circulation, okay? There's no point in sending blood to the lungs in the pulmonary circulation at this point because the lungs aren't filled with air. They're filled with amniotic fluid, okay? So there's no point in wasting blood on doing a task that it can't do in the lungs. The liver is not fully developed at this point, and so there's no point in sending blood through it yet, okay? And then at birth, what happens is all of these close up, and that restores the normal flow of blood in the adult so that the right side of the heart deals with oxygen-poor blood while the left side deals with oxygen-rich blood, okay? So we form the ligamentum teres. We are going to form the ligamentum venosum from the ductus venosus. The foramen ovale will close, generating the fossa ovalis, and the ductus arteriosus closes and forms the ligamentum arteriosum. If this does not happen, and occasionally this fails to happen, you're going to get mixing of oxygen-poor and oxygen-rich blood, and that's going to reduce cardiac output, and that will basically slow development, okay? The fetal period is weeks 9 through 38, where we see rapid growth of body structures in the embryo. You can see the embryo here at week 7, about 17 millimeters long, okay? You can see the placenta, and you can see the yolk sac, okay? You can see the fetus at the 3rd month, okay? It's just the end of the first trimester, okay? About 6 centimeters long. Here we are in the 5th month, about 19 centimeters long, okay? And the events that take place are listed here. I won't go through each one of these, okay? But this is nice to review. This is table 28.1 in your book, okay, or in the online resource, all right? Continuation of the table, okay? Now, the effects on the mother, okay? Okay, now the mother is going to go significant anatomical changes during this period as well, due primarily to the growth of the embryo and fetus and the effect of hormones on various tissue targets. Reproductive organs become engorged with blood. The vagina develops a purplish hue called Chadwick's sign. The breasts enlarge and the areola darken. The purpose of this ostensibly is to guide the infant to the source of the milk. Pigmentation of facial skin may increase, and we call this chloasma. The uterus expands and occupies most of the abdominal cavity. The ribs will flare, the thorax will widen, and you may end up with lordosis because you're now increasing the weight in the lumbar region. So what you're going to do is thrust the lumbar spine forward because of the change in your center of gravity, and this can result in lower back pain. Okay, relaxin causes the pelvic ligaments and pubic symphysis to relax and ease birth passage, and this sometimes leads to joint problems all over the body. And you end up with a weight gain of around 30 pounds on average. Good nutrition is critical. Average pregnant mother needs 300 additional daily calories. Multivitamins and folate reduce the likelihood of neurological problems such as spina bifida and anencephaly, which is lack of a formation of a brain, as well as spontaneous preterm birth. You can see how the changes take place. Here's prior to conception. There's 4 and 7 and 9 months. Now, one of the things that's occurring as a result of the expansion is that you're displacing the lower GI and you're pushing up on the diaphragm. So this can lead to indigestion, and this can also lead to a reduction in your vital capacity. Okay. Placental hormones include human placental lactogen and human chorionic somatotropin. The lactogen is designed to induce milk formation in the breast tissue. Okay. This somatotropin is going to promote fetal growth and glucose sparing in the mother, so that most of this goes for growth of the fetus. Parathyroid hormone and vitamin D levels are high throughout pregnancy because we need the vitamin D for adequate mineralization of fetal bone, as well as the other processes that rely on calcium in the developing embryo and fetus, such as muscle contraction, nerve transmission, and cell signaling. Okay. The GI tract will be affected. You'll often see morning sickness, which is thought to be due to elevated levels of human chorionic gonadotrophin, as well as high levels of estrogen and progesterone. Heartburn and constipation are common. Urinary system increases urine production because of the increased maternal metabolism and fetal waste. Okay? And you get frequent urgent urination because of stress incontinence on the bladder, because the expanding uterus is over the top of the bladder and producing pressure on it. Okay? The respiratory system may exhibit nasal edema and congestion. Your tidal volume will go up, but you may have difficulty breathing later in pregnancy, which we call dyspnea. Your vital capacity will also drop a bit. Cardiovascular system will see a blood increase of approximately 35%. This safeguards against blood loss during childbirth. Cardiac output goes up by close to 40%, and that moves more blood around the body. The venous return from the lower limbs may be impaired, and that can result in varicose veins, which are the result of incompetent valves in the lower extremities collapsing, causing the bulging vessel. Now these are a clot risk. Okay. Preeclampsia is the result. It results in insufficient placental blood supply. The fetus gets starved of oxygen. The woman becomes edematous and hypertensive, and we'll see protein in the urine. And we think this may be due to the mother's immune system actually attacking the fetal tissue. Okay? Remember that there are antibodies that are small enough to cross the placental barrier, and in some cases, they may end up producing these types of complications. We correlate this with the number of fetal cells that enter the maternal circulation. That makes sense because that will trigger an immune response in the female, and those antibodies can then cross the barrier, especially the IgG type. Parturition is simply giving birth. Labor are the events that push the infant out of the uterus and through the birth canal. Increased production of surfactant occurs in the weeks prior to delivery. You end up with an inflammatory process in the cervix that causes it to soften in preparation for labor. The fetus will determine when it's going to be born. During the last few weeks of pregnancy, what will happen is that the adrenal cortex of the fetus will generate cortisol, and the placenta will convert that into more estrogen. That causes more oxytocin receptor production in the myometrium and makes the myometrium more sensitive to increasing oxytocin levels. We see gap junction formation between the uterine smooth muscle cells, and the high estrogen levels end up countering the effects of progesterone, and you can often get false labor, which we call Braxton Hicks contractions. Surfactant protein from the fetal lungs can cause softening of the cervix. Fetal oxytocin from the posterior pituitary causes the placenta to generate prostaglandins. The oxytocin and the prostaglandins are going to cause smooth muscle contraction in the myometrium of the uterus. Okay, so it's the kid that starts this off. Now we should point out that the maternal oxytocin will also play a role here. Due to the high levels of prostaglandins, the contractions become more frequent and more vigorous. Antiprostaglandins, as a result, should not be used during labor. Increasing cervical distension is going to send nerve signals to the hypothalamus. That will cause more release of oxytocin from the posterior pituitary, resulting in a positive feedback mechanism where the more stretching of the cervix takes place, the more oxytocin is released, the more the baby is pushed through the cervix into the birth canal. And this ends once the baby is out of the birth canal and the act of parturition is over, and then the oxytocin bursts will cease. So you can see here how this all begins, right? We have estrogen that's derived from cortisol that goes into the placenta from the baby's adrenal cortex. That causes oxytocin receptors in the uterus. We see the formation of gap junctions in the myometrium, okay? And then oxytocin from both the fetus and the mother stimulate the uterus to contract, and that causes the placenta to release prostaglandins, which produce more vigorous contractions, and the stretching produces positive feedback that results in more oxytocin release from the mother's posterior pituitary. From labor's onset to a fully dilated cervix is the dilation stage. The longest stage of labor, which takes about between 6 and 12 hours, is to allow that opening to get big enough to get the baby's head through. Now, this is assisted by the effect of relaxin on the pubic symphysis and the fact that the cranial bones can somewhat change their shape. Because of the presence of the fontanelles, the head actually takes on a slight jelly bean shape as it moves through the birth canal and makes it easier for the baby to pass. Initial weak contractions are about half an hour apart and about half a minute long. They get more vigorous and rapid. The cervix will efface and dilate to 10 centimeters. The amnion will rupture. This is called breaking your water. The amniotic fluid will be released, and then the head of the neonate will enter the true pelvis. From full dilation to delivery, you get strong contractions about every 2 minutes that are about a minute long. The urge to push increases in the absence of anesthetic. Okay, so if you don't have a if you haven't had an epidural, okay, the urge to push will increase. That's why women who've had an epidural need to be coached to push more. Crowning occurs when the largest part of the head distends the vulva. Sometimes, if the head is too big, we have to do an episiotomy, and so what we do is we cut you from your the perineum in the region between the perineal raphe and the anus and the opening of the vagina. We have to make an incision in order to open up the pathway even more to get the baby's head out. Okay, and that reduces tearing. The vertex position is defined as head first. The skull dilates the cervix. Early suctioning allows breathing prior to complete delivery. Breech position is buttock first. Delivery here is more difficult. Often we use forceps or a C-section to deliver it through the abdominal wall, and this is because of the danger of the umbilical cord wrapping around the baby's neck and strangling the baby. So you can see the stages of birth here, right? Early dilation, okay? Late dilation at 10 centimeters. Expulsion, again, triggered primarily by the effect of oxytocin on the myometrium. And then placental stage as we push out the afterbirth. Strong contractions continue after the baby's out, and that causes the placenta to be torn from the endometrium. It also leads to compression of uterine blood vessels to limit bleeding. The afterbirth occurs about half an hour after the regular birth. All the placental fragments have to be removed, or else postpartum bleeding can take place. Okay? So let's take a look at parturition.
Labor has 3 stages. In the first stage of labor, the body prepares to give birth. There are 2 phases, early and active. Early labor can last from hours to days. First-time moms may spend more time in this phase. The cervix, or the opening to the uterus, begins to soften, efface or thin out, and dilate or open. The cervical changes are necessary so the baby can pass through. Usually, a woman's water breaks during the first stage of labor. Contractions or tightening of the uterus becomes strong and regular. Typically, the woman will have a 30 to 70 second long contraction every 5 to 20 minutes. There may also be a stringy, bloody discharge from the vagina called bloody show. This is considered normal. Active labor occurs when the cervix starts to dilate more quickly. Contractions get stronger, last longer, and occur more often. The cervix continues to open wider to about 10 centimeters. Then pushing can start. In the second stage of labor, the child is born. A healthcare provider usually encourages the woman to push to help the baby move down the vagina. She might push for minutes or hours. Once the baby's head starts to come out, called crowning, a provider guides the rest of the baby out. Childbirth is finished when the baby is completely out of the vagina. The provider then cuts and clamps the umbilical cord which connected mom and baby during pregnancy. The last stage of labor involves delivery of the placenta or afterbirth. During pregnancy, the placenta supplies food and oxygen to the fetus. Once the umbilical cord is cut, the placenta has to come out. Contractions typically begin 5 to 10 minutes after the baby's birth. During this time, the placenta detaches from the uterus. A provider may encourage the woman to push, just as she did to deliver the baby. After 5 to 30 minutes, the placenta comes completely out of the vagina. Then the process is complete.
Okay. Once the infant has passed through the birth canal, physiological adjustments in the infant prepare it for life outside the womb. The neonatal period is a 4-week period immediately after birth. The physical status is assessed about 5 minutes postnatal. An APGAR score of 0 to 2 points is given for heart rate, muscle tone, respiration, reflexes, and color. A score between 8 to 10 is considered healthy. Anything below that is essentially a danger sign. Okay? The first breath is caused by an increase in CO2 levels in the infant, which leads to central acidosis, and that stimulates the brainstem to trigger the first inspiration. This is often assisted by spanking the infant. And this requires tremendous effort because the airways are tiny and the lungs are collapsed. Surfactant in the alveolar fluid will reduce the surface tension so that the lungs can expand more easily. And this is one of the reasons why premature infants sometimes suffer from IRDS, which is infant respiratory distress syndrome, okay? Because they lack sufficient surfactant. The respiratory rate is about 45 breaths per minute for the first 2 weeks and then drops after that. And that just simply reflects the high metabolic rate that goes with tremendous growth. Keeping the lungs inflated is difficult for a premature infant, which is less than 6 pounds at birth. Surfactant production occurs in the last months of prenatal life, and so preemies need the surfactant given to them as an inhalant in order for them to avoid IRDS. There's a transitional period that runs about 8 hours after birth, where we see alternating periods of activity and sleep. Vital signs are irregular. The baby gags frequently as it regurgitates mucus and debris. It stabilizes with waking periods occurring between 3 and 4 hours. We've already talked about the changes that take place between the fetal and the neonatal circulation. The fact is that once we're out of the womb, we don't need the plumbing that allows us to get oxygen from the mother's blood supply and nutrients from the mother's blood supply anymore. So we can shut all of those modifications. The umbilical arteries and vein constrict and become ligaments. The proximal umbilical arteries become the superior vesical arteries to the urinary bladder. The distal umbilical arteries become the medial umbilical ligaments. The umbilical vein becomes the round ligament of the liver, which we call the ligamentum teres. The ductus venosus becomes the ligamentum venosus half an hour after birth. And pressure changes also cause the ductus arteriosus to close, forming the ligamentum arteriosum, and the foramen ovale to close, and that forms the fossa ovalis. Okay? Lactation is just the production of milk by the mammary glands and happens towards the end of pregnancy. Placental estrogens, progesterone, and human placental lactogen are going to stimulate the hypothalamus to generate prolactin-releasing factors. The anterior pituitary kicks out prolactin, which targets the breast tissue and causes milk production after the production of something called colostrum. Okay, so initially the colostrum is a protein-rich secretion high in antimicrobials and antibody and protein, and has a yellowish cast to it, and milk production happens about 3 days afterwards. So that's the colostrum. Okay. The IgA in the colostrum, as well as in the breast milk, is a form of passive immunity that protects the infant against infection while its own immune system is being established. And this is extremely important. Otherwise, it's very easy for an infection to set in while the infant's immune system has yet to fully deploy. This is released for the first 2 to 3 days, and that's followed by breast milk. Prolactin release drops prior to birth, but the lactation is sustained by the act of nursing. Okay? The nursing causes afferent impulses in the hypothalamus to cause prolactin release, which stimulates milk production for the next feeding. It also promotes oxytocin production, which causes the milk letdown reflex. And this is sometimes also triggered by the cry of a baby, okay? And that's the milk ejection. So oxytocin is milk ejection, prolactin is milk production. So there's the flow chart, right? And this is why we're called mammals, right? We use the mammary glands to produce milk in order for the baby to have nourishment so that it can be healthy, okay? Advantages of breast milk: fats and iron are absorbed more easily, amino acids are easily metabolized as compared to formula. You also have the IgA and the complement and the lysozyme and the interferon in there, as well as lactoperoxidase that protects from infection. Interleukins and prostaglandins prevent an inflammatory response that is too enhanced. Okay? Glycoproteins detect ulcer-causing bacteria and keep them from attaching to the stomach mucosa, okay, by eventually basically coating them. Natural laxative effects help eliminate the meconium, which is going to prevent physiological jaundice. Okay, so basically, this is bile buildup that is the result of the fact that the baby has a liver that isn't really working very well while it's in the womb. Okay. This also encourages bacterial colonization of the large intestine. That's where the E. coli and other species of beneficial bacteria take up residence. Women nursing half a year lose bone calcium, and it's replaced after weaning if she's on a healthy diet. Women can ovulate when nursing despite the inhibition of gonadotropin releasing hormone and the gonadotropins, although it's less likely. In the event that natural conception isn't possible, we can engage in reproductive technology in order to promote childbirth. Surgical removal of the oocytes under the influence of hormones that are super ovulators allows these oocytes to be harvested and to be fertilized outside the womb, and then we can return those fertilized oocytes to the woman's womb. The problem is it's expensive. It can be painful for the oocyte donor unless the oocyte donor is the woman herself. I mean, sometimes the reason for inability to fertilize is due to low sperm count in the male. Okay, and then another complication is that generally you do more than one fertilized oocyte in order to up the likelihood that you'll get an effective implant in the uterus, and this can raise the possibility of multiple births. Okay, and so they give you the option of selective reduction, which is basically an abortion procedure. In vitro fertilization is, as we've described, taking place in a container outside the woman's body. Okay, the blastocysts are transferred to the uterus for implantation. We also have zygote intrafallopian transfer, where fertilized oocytes are placed in the fallopian tubes, and gamete intrafallopian transfer, where sperm from the male and oocytes from the female are put together in the fallopian tubes so that fertilization is more likely. So basically what's happening is that we are removing some of the obstacles to fertilization that a normal sperm cell encounters on its way to the egg. Cloning is a different procedure altogether. As far as we know, it hasn't been done in humans. Essentially what you do when you clone is you enucleate the egg, and then you introduce the genetic material from another individual, from a body cell, and what you generate then is a clone of that individual, has the exact same genetic composition, and as a result would be very similar to the donor. Okay, it's been done with sheep, it's been done with rabbits, it's been done with salamanders. It has not been done, as far as we know, with humans. Okay. All right. What follows is a walk through everything from fertilization to birth, It's a very interesting video called The Miracle of Life. So I encourage you to stay tuned and to watch. A lot of fascinating stuff in this video. And this brings to an end the podcasts for this term. Again, if you have any questions, make sure and get in contact with me by email or cell, and I should respond to you within the day. I thank you very much for listening.
The following film, The Miracle of Life, is about the process of human reproduction. Please use discretion when choosing to view the program. Four and a half billion years ago, the young planet Earth was a mass of cosmic dust and particles. It was almost completely engulfed by the shallow primordial seas. Powerful winds gathered random molecules from the atmosphere. Some were deposited in the seas. Tides and currents swept the molecules together. And somewhere in this ancient ocean, the miracle of life began. The first organized form of primitive life was a tiny protozoan. Millions of protozoa populated the ancient seas. These early organisms were completely self-sufficient in their seawater world. They moved about their aquatic environment, feeding on bacteria and other organisms. They were covered with hundreds of tiny whipping hairs called cilia and flagella that made movement possible. From these one-celled organisms evolved all life on Earth. And the foundation of life, the cell, has endured unchanged since the first tiny organisms swam in the cradle of life, the sea. Every living being is made up of cells, the basic units of life on Earth. And all cells reproduce themselves by dividing into two identical cells. This is a single body cell from a human being. And this is the process by which all cells reproduce. Now, there are two cells, each exactly like the parent. In the cell's nucleus is the extraordinary chemical substance DNA. The DNA is contained in 46 chromosomes in every cell of the human body. Each chromosome, in turn, is made up of thousands of genes, discrete segments of DNA which lie along the chromosome. Penetrating even deeper into the structure of the chromosome, we see the DNA molecules themselves. DNA contains all the genetic information of the cell, and it is the only living substance capable of reproducing itself exactly. Without DNA, duplication, and therefore life itself, is not possible. All DNA in all living organisms is chemically identical, but its arrangement in genes and chromosomes determines what the cells will become. Here, a tiny primitive organism, the protozoan. A massive complex mammal, the elephant. A palm tree swaying in the tropical breeze. Or a human being. We are about to witness, for the first time, a wondrous and complex process. The actual conception and growth of a new human being. Each beginning lies deep inside the mother's body, in the ovaries. Each ovary contains 250,000 egg cells, which reach full development even before the woman herself was born. The eggs each contain 23 chromosomes, the mother's genetic contribution to her future offspring. During each menstrual cycle, the ovaries produce hormones which stimulate the growth of a single one of these eggs. At the midpoint of her cycle, the follicle, which encloses the egg in its protective layers, ruptures. This is ovulation. The egg travels through the fallopian tube, which connects the ovary and the uterus, or womb. In the tube, the egg waits for sperm from the father to fertilize it. The fertilized egg, now with genetic material from both parents, moves through the tube to the uterus, where it attaches itself to the nutrient-rich lining. And here, the ovary itself. In cross-section, stained blue for better visibility, we see the hundreds of tiny egg cells. This one is nearly mature and is surrounded by its nutritive layers. When it is fully developed, the follicle which encloses the egg will swell, like this one, with fluid, the liquor folliculi. Inside the follicle, the egg and its nutritive layers floats in this water of life, which has the same salt content as the sea. The swollen follicle here on the left is then gently brushed by the swaying fimbriae, the outermost fringes of the fallopian tube. These fringes are activated by the hormones just before ovulation. They sweep over the surface of the ovary, searching for the newly released egg. As ovulation nears, the fringes become filled with blood. The follicle at the center is about to burst. Directly above the white reflections, the liquor folliculi pours out of the follicle. Concealed somewhere within it is the egg. The fringes search for the newly released egg. It lies here, surrounded by a cloud of its own nutritive cells. It is only the size of a grain of sand. the far right, the egg is drawn into the fallopian tube. Deep inside the egg is the nucleus of new life itself. This is the mouth of the fallopian tube. Its almost imperceptible muscle contractions move the egg along toward the uterus. Here, the egg has reached the interior of the tube itself. These many folds of tissue are lined with tiny cilia, which maintain a constant, gentle motion that draws the egg along its length. The inside of the tube is actually only about twice the thickness of a single human hair. It takes the egg 3 to 4 days to travel this distance of only 5 inches. The translucent egg here lies in a muscle fold deep inside the fallopian tube. On the diagonal edge below the egg, we can see a thin, shimmering layer of the cilia which line the tube. Gently moving the egg along, they are exactly like the cilia of the primitive protozoa. The egg must join with a sperm within 24 hours of leaving the ovary for conception to take place. If no sperm are present, the egg disintegrates, and the same cycle will happen again the following month and throughout all the woman's childbearing years. This remarkable cycle is made possible by complex molecules, the sex hormones. Crystallized and stained, they look like this. Hormones control a woman's entire reproductive cycle. Hormones control a man's reproductive ability as well. Only 7 weeks after conception, the fetal system is permanently sensitized to be male or female by the sex hormones. One fetus will develop ovaries, and its brain will be programmed to release hormones in cycles. It is female. Another fetus will develop testicles, and its brain will release constant low-level amounts of sex hormones. It is male. "77 billion human beings have lived on this planet. 4 billion live here now. And every year, 128 million new ones are born. We have already seen part of the woman's remarkable contribution to this unending process. Now, we will explore the man's. During a man's lifetime, his testicles produce billions of sperm. They are stored here in the epididymis. The Cowper's gland produces a lubricating fluid to aid the sperm's journey. The prostate gland secretes an alkaline fluid to protect the sperm. Together, they produce most of the fluid called semen. During ejaculation, the sperm leave the epididymis, moving through the vas deferens, this tube in the body cavity. They approach the seminal vesicles, which release a solution of sugar to nourish them. Cowper's and prostate fluids combine with the sperm, and the blended semen continues out of the man's body through the urethra in the penis. This is the interior of the urethra itself. We are traveling up into the man's body from the outside through the penis. The red areas are blood vessels. The black spots are calcium deposits. In reality, the journey is only 7 or 8 inches long. Ahead, the red area is the opening to the bladder. During sexual arousal, a valve here stops urine from entering the urethra. The valve is now open. Here, near the bladder, the urethra passes through the prostate gland. The walls of the prostate have more than 30 orifices, like these, which squeeze out prostate fluid when sperm pass through the urethra. It is about the size of a golf ball, and it resembles a sponge. Inside, it is filled with the small cavities which produce the fluid. These are the testicles, highly magnified and without their usual protective pouch. Unlike the ovaries, their counterpart in women, the testicles lie outside the body cavity. They are made up of the tightly coiled seminiferous tubules, which have a total length of 700 feet. In the tubules, sperm are produced at the extraordinary rate of 100 million every 24 hours. In the tissue between the tubules, the sex hormone testosterone is produced. The testicles are the essential male organs of reproduction. They function well only under very specific conditions. One of these specific conditions is temperature. Sperm are produced most efficiently at several degrees below normal body temperature. These images from a thermal camera reveal the various temperature zones in the scrotum, the pouch of skin in which the testicles are suspended. The white areas are the warmest, and the green ones the coolest. The scrotum keeps the testicles away from the body's heat. This is a cross-section of one of the tubules in the testicles, stained blue and magnified 2,000 times. At the dense center of the tubule is the transport canal, which carries mature sperm away to the epididymis for storage. The smaller compartments produce many new immature sperm every 36 hours. As the sperm become more mature, they move closer to the central canal. The average man produces over 400 billion sperm in his reproductive lifetime. These sperm, magnified 4,000 times, have been in production for approximately a month. They are tightly packed in the tubule within the testicle, their heads and tails intertwined. Inside the developing sperm heads, the normal body cell's 46 chromosomes have been reduced to 23. But this reduction of chromosomes, so essential to future reproduction of the species, puts the sperm in mortal danger. The man's body considers these cells enemies. But the sperm are defended by nurse cells, like this one, and the large cell at the right. They form special protective barriers around the maturing sperm cells. Each nurse cell cares for many sperm at a time. This tendril is part of the remarkable communication network which connects each nurse cell to over 150 separate sperm cells throughout the tubule. The tendrils enable the nurse cells to feed and protect the maturing sperm and to move them closer to the transport canal. This sperm is nearly mature. During its development, it has lain in total passivity in the tubule, being protected and nourished by the nurse cell. When mature, like this one, it is transported by the nurse cell to the epididymis. Millions of sperm are stored in these densely packed tubules, which, if stretched out, would be 15 to 20 feet long. The mature sperm in the epididymis will pass out of the man's body through ejaculation 200 to 300 million at a time. Or they will eventually die and be reabsorbed. These sperm carry the man's genetic material, and they are fully mature. But they are not yet able to fertilize a woman's egg. They do not attain that ability until they are actually far up inside the woman's body. They may stay in the epididymis, the bag-like structure on the left, for several weeks. While here, they develop the ability to swim. A sperm propels itself by means of a long tail, a flagellum, which lashes vigorously from side to side. This extraordinary activity has but a single purpose: to deliver the genetic material in the sperm's head quickly and precisely to the woman's egg. On the tip of the sperm's head is a layer of enzymes and enzyme inhibitors. The inhibitors preserve the enzymes, which will be needed later to penetrate the surface of the egg. This is how the sperm's head looks without its enzyme cap. The material of the head itself is loaded with the actual genetic information which will be transmitted to the egg. These are the strands of DNA which will be transformed into chromosomes. This is one of the many fuel packets arranged along the midpiece of the sperm. The fuel consists of sugars, which is converted into energy for the sperm's locomotion. The propulsion system of the sperm's tail is made up of a bundle of cords or filaments. The filaments are covered with thousands of tiny hooks. As the hooks intermesh, the whole bundle begins to bend, and the tails of the sperm begin to whip around like this. It is a primitive but highly efficient system of propulsion. The system does not function perfectly every time. On the average, up to 20% of a normal man's sperm are deformed or imperfect in some way. Perhaps one reason why a man produces so very many sperm is that so very many things can go wrong with the sperm themselves. They may have 2 tails, like this one. Some even have 3 tails. In another common deformity, the heads of these sperm appear to be nearly severed from their tails. This sperm has been attacked by bacteria. And this one may have been produced too rapidly. It will never complete its development. White blood corpuscles, the large round cells, are often found together with defective sperm. Their presence indicates the likelihood of infection, fever, or even a common cold. Most of these sperm deformities could have been caused by just a slight elevation in the temperature of the testicles. Environmental conditions, too, often damage sperm production. Overcrowding, stress, smoking, chemical pollution of air and water, occupational hazards, radiation, poor nutrition, all part of modern life. Even our modern clothing may affect sperm production. Tight blue jeans, for example, hold the testicles up close to the body, perhaps raising their temperature those few critical degrees. But in spite of such obstacles, human beings have been successfully reproducing for millions of years. Since the first organisms swam in the sea, the cycle of life has been continuous, fueled by the need and the drive to reproduce. Among human beings, that drive is intimately linked with attraction and desire. Affection and romantic love may initiate the dance of courtship, which may lead to conception. Sexuality, affection, and tenderness can all be part of the complex rituals of mating. But desire and physical love possess a silent language all their own that signal partners' readiness to each other. The eyes are part of that silent language. Visual impressions stimulate and excite special nerve cells in the sexual areas throughout the body. The skin, too, is part of the silent language. It is amazingly sensitive and responsive to the touch. It contains millions of these sensory receptors, which, when stimulated, direct messages of sexual arousal to the brain. Many of these are concentrated in the body's most responsive areas, the erogenous zones. One of these, the man's penis, is densely packed with sensory receptors, here seen at enormous magnification. They relay messages of sexual stimulation to the brain, which triggers the complex sequence of events leading up to ejaculation. The length of the penis is made up of many small cavities called erectile tissue. When a man is sexually aroused, the signals from the brain cause these erectile tissues to become engorged with blood. As we can see in these images from the thermal camera, the sudden flow of blood to the penis raises its temperature. The man's blood pressure, heartbeat, and respiration increase. And the blood causes the penis to become erect and elongated so that sperm can be most easily delivered to the woman's egg. In the epididymis, the hundreds of millions of waiting sperm, which will soon be expelled from the man's body, accumulate in a mass. As sexual arousal reaches its peak, the entire male sexual system must function perfectly for ejaculation to take place. Cowper's gland, prostate, seminal vesicles, and epididymis. This is the actual journey made by the sperm. At the climax of sexual arousal, the man's nervous system triggers rapid involuntary contractions of the muscles in the walls of the epididymis. These propel the sperm up into the man's body along this tunnel-like tube, the vas deferens. The sperm themselves travel the distance, a little over 12 inches, in only a few seconds. The seminal vesicles release nourishing fructose, which blends with the sperm as they pass by. They mix with the prostate fluid. The blended semen approaches the urethra. Its total volume is actually about 1/2 a teaspoon. This is the last leg of the journey. The semen containing the sperm is forced out through the penis by the man's muscle contractions. And at last, it is ejaculated into the woman's vagina. As the semen enters the woman's body, it immediately slows down and coagulates. Perhaps it becomes thicker to ensure that most of the sperm stay within the vagina. Or perhaps the coagulation is a kind of defense, for the environment in the vagina is extremely acid. The acidity protects the woman against bacteria and infections, but it is also dangerously inhospitable to the sperm. Nearly 1/4 of them will die almost immediately. About 20 minutes after ejaculation, the semen becomes fluid again. The surviving sperm become vigorously active, swimming rapidly against the downward currents in the woman's body. There is great urgency, for the sperm will remain viable, able to fertilize, for only 28 to 48 hours. This is the sound made by thousands of active sperm. The sperm's great activity has but one goal: to find and fertilize the egg. There are so many of them that success might seem assured. But the barriers, too, are numerous. The high percentage of defective sperm has already lowered the odds. So has the vagina's hostile acid environment. And the woman's own defense system attacks the sperm. They are unwelcome cells from another organism, and they are potential enemies. Her defense cells aggressively protect her from the invaders by destroying them. The sperm are programmed to seek the egg, but some of them try to fertilize the first round object they find. In this case, an ordinary body cell. Still, in spite of the obstacles, hundreds of thousands of sperm make their way up through the vagina to the cervix, the mouth of the uterus, which will lead to the fallopian tubes and the egg. The cervix secretes strands of a special fluid protein called mucin. During ovulation, it is very liquid and easy for the sperm to swim in. The strands of mucin provide tiny channels less than 1/100th of a millimeter wide, directing the sperm closer to their objective. But many of them never find the strands of mucin, and they will die in the vagina. Those that survive will swim up the mucin channels toward the uterus. They swim in dense bunches. The outer ones may protect the inner ones from the woman's acidity and defense systems. Once inside the cervix, the sperm continue on their journey. Some remain here, perhaps to provide a backup for those that continue on. The rest proceed up through the cavity of the uterus in search of the egg. Their number is noticeably reduced. Here, from inside the uterus, we see the openings to the two fallopian tubes. The sperm swim toward them. One leads to the waiting egg. The other to an empty tube. Only half of the few remaining sperm will swim up the tube which holds the egg. The tubes are lined with millions of tiny cilia, the same cilia which helped draw the egg from the ovary toward the uterus at ovulation. These cilia sway constantly, creating a gentle downward current that the sperm must swim against. Some sperm get stuck in among the cells lining the walls of the tube. Others lose their sense of direction. During this part of the sperm's journey, the layer of enzyme inhibitors at the tips of their heads is slowly being worn away. The sperm are fully capacitated and able to fertilize the egg, if they ever encounter it. Of the 200 million sperm that begin this long journey, only about 50 ever reach the egg. The egg is surrounded by 2 layers of the nutritive cells which have nourished it on its journey in the fallopian tube. The sperm immediately release their digestive enzymes to break through these protective nutritive layers in order to reach the egg itself. At best, only one will eventually enter and fertilize the egg. The joint force of their exertions starts the egg rolling around like a mysterious celestial body. All the while, the sperm are dissolving their way through the egg's outer layers. The first sperm to reach the egg's membrane is immediately drawn inside. This is the tail of the penetrating sperm seen from the surface of the egg at enormous magnification. And this is a picture from inside the egg itself. It has been fertilized now for only a fraction of a second. Here is the penetrating sperm with a circle around it from a wider perspective. Only this single sperm will be allowed inside the egg. A rapid biochemical change took place in the egg's membrane. It is now impermeable to all other sperm. Once inside, the sperm too undergoes a dramatic transformation. It loses its midpiece and tail. The tailless head swells up, almost like rising bread. This hole in the sperm head's covering is the first indication that it is about to rupture and release its precious genetic material. Here, the first strand of genes is making its way out of the sperm into the egg. The genetic material continues to spill out from the sperm head. Its tightly packed molecules contain the father's hereditary message. They almost seem to have been dispersed by an explosive force. This is an extreme close-up of the genetic material itself. The magnification on the television screen is over 500,000 times. the genetic material of the mother's egg and the father's sperm combine. Two cells have joined to make a single new cell. And within 24 hours, that new cell begins to divide. These early cell divisions of the human embryo have never before been filmed. The fertilized egg now has two nuclei, the small indentations at the center of the cell. The first division of the egg is beginning. Now there are two identical cells, still within the same nourishing material of the original egg. No growth has occurred, but rather a distinction between cells. Once it has begun to divide, the fertilized egg is called a zygote. Now there are 4 cells. The divisions happen at such an accelerated pace that there's little opportunity for the new cells to grow before they divide again. Now there are 8 cells. Each new generation of cells in the zygote is smaller than the one before it. Another division, and another. As the zygote divides, it moves along the tube toward the uterus. Now it has become a dense, compact cluster of many cells. And after 5 days, it is called a blastocyst. It is still no bigger than the original egg. Its center is filled with liquid. Within 10 days of fertilization, the blastocyst implants itself firmly in the lining of the uterus. Already, the mother's hormones are directing changes in her body to prepare it to support the growing embryo. Barely visible to the naked eye, the embryo will be nourished by the lining of the uterus and then by the placenta. After 2 weeks, the embryo is elongated. It is barely 1/10 of an inch long. At the top, what will become its head and brain. Below, at the tail, the embryo is firmly attached to the placenta, which will nourish it. At 4 weeks, the embryo has arm buds and is distinctly curled. It has the beginning of eyes. At 5 weeks, the nose begins to take shape. At 6 weeks, leg buds are apparent. The embryo is less than 1/2 inch long. It floats inside the fluid-filled amniotic sac. Its spine is clearly visible. At 7 weeks, the embryo is 3/4 of an inch long. It can move its hands, on which there are clearly defined fingers. Its internal organs are visible. The eye lenses are formed. The skull bones, rich in blood vessels, are growing together at an angle at the crown of the head. At 8 weeks, the fingers of the hands are well defined, and the toe joints of the feet are clearly visible. At about 10 weeks, the embryo is considered a fetus. It can move actively. There is a suggestion of an ear. It is 2 inches long, and it still has the stump of a tail. At 11 weeks, it is 2.5 inches long. At 12 weeks, 3 inches long. Here, the umbilical cord, connecting the fetus to its food supply. By 14 weeks, it can bring its hands together and suck its thumb. By 15 weeks, the sensory organs are nearly completely formed. And by 16 weeks, it is actively turning inside the mother. This fetus is 18 weeks old. It is 5.5 inches long and is here shown 15 times larger than its actual size. Its mouth and lips are fully formed, and it has the strange nasal plugs whose purpose is not yet understood. The eyes of the fetus are closed, but it can see. This sound is made by the fetus as it breathes in the amniotic fluid in what is known as fetal respiration. It brings the fluid in through its mouth and then breathes it out again. The umbilical cord is the fetus's link to its source of life, the mother. Here are the fetus's sex organs. All its important physiological systems have developed, but it will be at least another 8 weeks before the fetus has even a remote chance of surviving outside its mother's womb. Whatever signals the beginning of birth is still a mystery. But when the fetus is ready to be born, the uterus begins its powerful contractions, and the process of birth begins.
Pull your legs right in. Keep it coming in. Pull them right in. Keep it coming. Take another quick breath. Hold it. Push. Don't stop. Keep it up. Hard. One more in. Deep breath, quick. Push. That a girl. It's so close. Yeah. I wish you could see this. Can you see this, baby?
Now stop pushing.
Stop pushing. Blow candles. Blow candles.
Oh,
Beautiful job.
Here he is, gonna suck out his little nose and mouth so we can take some nice, clean first breaths. Nothing's little about this baby. It's a stomach. It's another girl. It's another girl. Oh, she's so
Happy birthday.
Shop online for the best of public television and radio. Shop WGBH. Go to shop.wgbh.org. Every purchase directly supports your favorite shows on WGBH, and members always get a 15% discount.